Okay, so this is the third screencast, and what this is the beginning of the components of a nerve impulse. And we're going to start off with what happens when a nerve impulse is not being conducted. All right, electricity is still going back and forth across neurons, and that we refer to as your resting membrane potential or your resting potential. So on the other side of this animation, we'll discuss it a little bit further. Okay, here, listen up.
When solutes such as ions are placed in a solution without physical barriers, they become evenly distributed throughout the solution by a process called diffusion, the random movement of particles toward a state of equilibrium. For the mixture shown here, equilibrium is reached when the concentration of each ion is evenly distributed throughout the solution. Neurons, like all living cells, are surrounded by a plasma membrane that is impermeable to ions. This property allows a neuron to maintain different concentrations of ions between the inside and the outside of the cell by preventing the passive diffusion of ions from regions of high to low concentration. In a typical neuron at rest, there is a large difference in the concentration of ions such as sodium and potassium between the intracellular and extracellular environments. In addition, the interior of the cell has a high concentration of protein ions, each with a considerable negative charge. The plasma membrane is composed of a lipid bilayer. The hydrophobic interior of the bilayer prevents the movement of ions across the membrane. The only way that ions can diffuse across the membrane is by passing through specialized channels that permit the movement of particular ions while excluding others. These channels can be in an open or closed state. When the neuron is at rest, most ion channels are closed. However, a few potassium channels are open, and potassium ions are therefore free to diffuse out of the cell into the extracellular environment. Note that channels can be highly selective. The potassium channels do not permit the passage of sodium ions. Since only a few sodium channels are open at rest, very few sodium ions flow into the interior of the neuron. If we consider only the potassium ions, which are relatively free to cross the membrane, we notice that the intracellular concentration remains much higher than the extracellular concentration. This is because the potassium ions are pulled by two different and opposing forces. First, a diffusion force acts to pull potassium ions down their concentration gradient toward the exterior of the cell. The movement of potassium ions out of the cell results in a depletion of the internal positive charges, causing the potential to become increasingly negative. An electrical force produced by attraction of the positively charged potassium ions toward the negative cell interior generates an electrical gradient that pulls potassium back into the interior of the cell. The diffusion and electrical forces eventually come into balance, and an electrical potential is achieved at which the electrical gradient is exactly balanced by the diffusion gradient. This potential is the equilibrium potential for potassium. At this electrochemical equilibrium, there is no net movement of potassium ions. On average, for each potassium ion that leaves the cell, another returns. The electrical potential across the membrane can be measured using a pair of electrodes. An oscilloscope is used to continuously display the potential difference between the reference electrode and the recording electrode. When both electrodes are in the back outside of the cell, no difference in potential is measured between them, and the display registers 0 millivolts. However, when the recording electrode enters the cell, the display records a membrane potential of about negative 60 millivolts. This value represents the resting membrane potential of the neuron, the difference in electrical potential across the membrane during an inactive period.
Okay. So that is that animation. Let's bring up the picture that we're going to use. All right. So again, this picture is going to be all over the place here. All right. So to the top picture there, all right, this is going to be one that we're going to draw in parts into our diagram notebook. Picture A represents a resting membrane potential. All right, so neurons, as you heard, pump sodium and potassium ions across their membranes to generate a resting potential. Neurons generate and move They use the word conduct electrical signals by pumping positively charged ions, both sodium and potassium, okay, across the membrane. And what happens is that the unequal distribution of the ions, there's more on one side than the other, creates a difference. And if there's more positives on one side, that means the other side is negative with reference to that side. And the resting membrane potential is that difference. It's the difference in charge across the membrane when the neuron is not under an impulse, not firing an impulse. Okay? And although you heard the resting membrane potential on the animation as negative 60 in IB in most of the world, and we talk it, so in a typical resting potential, the inside of a neuron, okay, the inside of a neuron is more negative relative to the outside of a neuron, more relative, and that is approximately negative 70 millivolts. So it's going to take you through a couple of the notes here that we have. So all I have is that picture, the animation, and the notes. All right. So not conducting an impulse, not firing is the word that they use. All right. And what we need then is to keep an electrochemical gradient with a negative charge inside the neuron, inside the on one side of the membrane of the neuron with reference to the other. And that allows the gradient allows the ions to move back and forth. Come on. All right. So there we saw it. The inside is relatively negative. Now, what moves it? Okay. A channel protein moves it. So you notice I have in italics the specific channel. So it's a sodium potassium pump. And that's an important thing because there are other channels coming up. So sodium potassium pump maintains resting membrane potential. All right. So it's controlled by that. All right. So it is ATP dependent. And so pump is where's that there? The pump is a reference word for active transport. So active transport, ATP dependent sodium potassium pump. All right. What happens? Sodium is transported out, potassium is transported in. But how do we keep the difference between positive outside, negative inside? Because it expels 3 sodium for every 2 potassium that goes into the cell. Okay. Additionally, though, I think I put it in there that the membrane is more permeable to potassium. So additionally, some potassium ions leak out of the cell as well, adding to the positive outside portion of the cell. All right. Resting membrane potential is negative 70, and that results in the difference between the outside and the inside of the cell. All right. So resting membrane here. All the important information on here, resting membrane potential. And we conclude this screencast.